In this video, I will show how to align a posed ligand to a reference, in essence, a rigid body alignment. I will show this with Forge, but the same procedure is done in Torch. In some cases, you may want to align a ligand in its present 3D conformation to the reference. This may be the case when you want to compare a binding mode hypothesis from a field templater experiment to the actual binding mode from a protein-ligand crystal structure. In Forge, I've loaded the protein-ligand crystal structure for maraviroc in the CCR5 receptor. I've previously used field templater within Forge to calculate a set of binding mode hypotheses using maraviroc and some other inhibitors. I now want to compare the conformation of maraviroc from the field templater experiment to the actual binding mode from the protein-ligand crystal structure. I will right-click on the structure and choose Align Molecules. I will not do the conformation hunt as we're doing a rigid body alignment, but I will choose to do the alignment under normal conditions, deleting the existing alignments. When the alignment completes, I see that the field templater generated conformation of maraviroc is virtually identical to the actual binding mode, except for one end of the molecule where the functional group is rotated 180 degrees. Looking at the field point patterns alone, with the structures turned off, I see that the major field point associated with this group is consistent between both binding modes, and the hydrophobic field points have only very minor differences in size, and that corresponds to swapping of methyl to isopropyl. So this calculated binding mode hypothesis is actually quite good. If you have any questions or would like more information about doing rigid body alignments in Forge or Torch, please contact support@cresset-group.com.